Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow at Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Cell, the Unit of Life. So till now we have studied about the prokaryotic cells and several organelles of eukaryotic cells. In today's class we will discuss about the cytoskeleton and the nucleus part of the cell. So the first thing that we'll study is about cytoskeleton. So what is cytoskeleton? As the name suggests, cyto means cell and skeleton means framework. Okay, so the word cytoskeleton means that it is the framework of cell. So the cytoskeleton is a constituent of a cell that basically provides the framework to the cell. Okay, and if we talk about the nature of the cytoskeleton, so what is the nature of the cytoskeleton? Cytoskeleton is proteinaceous in nature. That is, it is made up of protein. So it is proteinaceous in nature. Okay. Secondly, if you look at the structure of the cytoskeleton, it looks something like this. So this green color here, it is representing the cytoskeleton. And this blue color here is representing the nucleus. And the outer red color is showing the plasma membrane. So it is a type of cell, and this is the overall shape of this cell. Okay. So what we can see here, if we look at this green structures here, what we can see that these green structures are looking something like a thread-like, very minute, right? So, and this thread-like structure is known as filamentous structure. So we can write that apart from it is made up of protein, it is also filamentous in nature. Okay, so it is filamentous in nature. Filamentous means thread-like. Thread-like. And what else we can write? That it is very minute, very fine, right? These are very fine threads. So we can write the structure is, or the nature of this cytoskeleton is very minute or fine. Okay. Now, if we talk about the function of the cytoskeleton, then what are the functions of the cytoskeleton? Because it is providing a framework, it is act as a framework of cell, right? So framework means it is providing a structural support to the cell, right? So the function we can write that it provides structural support. It provides structural support to the cell. Okay. The second thing, if it is providing structural support, it means it is also help in maintaining the shape of the cell. Because if you see in the diagram, these cytoskeletons are extended in such a way that they are somehow helping in giving this cell a particular shape. For example, if you see here, so this is the shape. And these cytoskeletons are extended in such a way, or they are stretching the plasma membrane in such a way that the cell is getting this shape, right? So we can say that it helps in maintenance of the shape. Maintenance of the shape of cell. Now the third thing we can write is it is also providing mechanical support. Providing mechanical support to the cell. And the fourth thing, it also helps in mobility. That we will see in later slides that it also helps in the mobility. One important thing about the cytoskeleton is that it is present only in eukaryotes. The cytoskeletons are present only in eukaryotes. So in prokaryotic cells, we have not studied anything about cytoskeleton, right? So these are present only in eukaryotes. Now, these cytoskeletons are of three types. So if we talk about the type of the cytoskeletons, there are three types. On what basis? On the basis of diameters. So on the basis of diameter of the cytoskeletons, there are three types. And the first is microfilament. The first type is named as microfilament. The second is called as intermediate filament. So as the name suggests, micro is very small. Okay, so these are microfilament. Then the second is intermediate filament. That has some intermediate size or diameter. And the third is microtubules. So it has some tubular-like structure. So it is microtubules. And if we talk about diameter, so this is in the decreasing order of the diameter. So it has the smallest dia. The first one has the smallest dia, and the last one has largest diameter. Okay, the last one is the largest diameter. So these are the three types of cytoskeleton. Now we'll study each of these types one by one. So the first type is microfilament. So in the microfilament, if we write about the diameter of the microfilament, so the diameter of the microfilament is 5 nanometer. Okay. Now, second thing, if we look at the structure of this microfilament, so if you look here the structure, this is the one structure. If you look, so what this structure is looking like, it is looking like rod shape. So the microfilaments are rod shaped. Okay. Thirdly, we are not seeing any branching here, right? The branching should be, otherwise it will appear something like this. So we are not seeing this branching. So we can write that microfilaments are unbranched. These are unbranched. Next, what they are made up of. So in the diagram, we can see that it has two components. The first one is filamentous actin. The first one is filamentous actin. The second is globular actin. So it is made up of, it is, so it is made up of a, okay, so it is made up of, it is made up of what? It is made up of a special protein, and the name of this protein is actin. This is actin. So it is made up of protein, and this protein is called as actin protein. Now, if we talk about the nature of the actin protein are contractile in nature. They are contractile in nature. So what do we mean by contractile? It can undergo contraction and relaxation, right? And hence these actins are major components of our muscle cells. Okay. Okay. Next, if we talk about this actin can be also of two types, as we see in the diagram. The one is called as F actin or filamentous actin. So F actin, or we can also call it as filamentous actin. And the second type is G actin, or we can call it as globular actin. So we can call it as globular actin. Okay. So these are two types of actin of which a microfilament is made up of. Now, if we talk about the function of these microfilaments, so the first function is they are found in close association with the plasma membrane. So suppose you have this cell, so this microfilament will be found somewhere very close to the plasma membrane. Okay. So because they are found in close association with the plasma membrane, so what they do, they provide support to the plasma membrane. So because of this reason, they provide support. They provide support to the plasma membrane. Okay. So the first function of this is they provide support to the plasma membrane. Secondly, they also help in amoeboid movement of the cell. So they also help in amoeboid movement of the cell. Now, what is amoeboid movement of cell? So amoeboid means amoeba-like movement, right? So we know one unicellular organism, that is amoeba, and it moves the way it moves is called as amoeboid movement. But apart from amoeba, we also have in our body a cell that also have this amoeboid movement, and that cell is known as macrophages. So the macrophages and amoeba, these are the cells that undergo this amoeboid movement, and this amoeboid movement is caused by this special cytoskeleton that is known as microfilament. Okay. Now, after this, the second type of filament is known as intermediate filament. Okay. So the intermediate filament, if we talk about the diameter of the intermediate filament, so the diameter of the intermediate filament is diameter is 10 nanometer. Okay, so if you talk about this diameter, so the diameter of intermediate filament is of 10 nanometer and it looks something like this. Okay, now what it is made up of? So as the microtubules, microfilaments we saw was made up of actin protein, the intermediate filaments are made up of, these are made up of acidic protein. One of the example is keratin. Okay, so these are made up of acidic proteins, and these proteins are non-contractile in nature. So unlike microfilaments, these are non-contractile. Just these proteins are non-contractile in nature. It means it cannot undergo contraction. Okay, so they are rigid. So these are non-contractile in nature. Okay, and where these intermediate filaments are present, these are present all over the cell, but they are mainly present or found around the nucleus. So intermediate filaments are found around the nucleus. Okay, they are found around the nucleus. So if you talk about the function of these filaments, so they provide new structural support to the nuclear membrane. Okay, they are found around the nucleus and hence they provide structural support. They provide structural support to the nuclear envelope or nuclear membrane. Okay, so this is all about the intermediate filament. Now, the third type of cytoskeleton is called as microtubule. So in microtubule, if we talk about the diameter of the microtubule, so the dia of the microtubule is the largest among the three. So the dia, it has two diameters. If we talk about the outer diameter, so this is the structure in which this is the outer diameter and this is the inner diameter. Okay, so if we talk about the outer diameter, so the outer dia is of 25 nanometer, and if we talk about the inner diameter of microtubule, it is 15 to 17 nanometer. Okay, now the second thing is about the shape. So if you see the figure, what we can write, we can see that it is of cylindrical shape, right? It is looking something like this, right? So it is cylindrical in shape. It is cylindrical in shape. And secondly, it is hollow, right? Because it has space in between. So it is cylindrical and hollow. Okay. Now the third thing, what it is made up of. So microtubules are made up of special proteins, that is tubulin. Okay, made up of tubulins. Now these tubulins are further of two types, alpha tubulin and beta tubulin. Alpha and beta tubulin. Okay. Now in case of microtubules, how these tubulins are arranged? So these tubulins are arranged in an alternate fashion. Okay. So suppose this is a alpha, then this is a beta, then alpha, then beta. So this arrangement is something like this. Okay, alpha beta. And this arrangement together makes a structure known as protofilament. This is known as protofilament. And 13 such protofilaments. So if you say, so 13 such protofilaments, this number will go like 13, and 13 such protofilaments make microtubule. Okay. So 13 protofilaments together, they roll and they make microtubule. So in simple words, what you can say that alpha and beta tubulin, they make protofilaments, and 13 protofilaments together, they make microtubules. Okay. That's why if you see the cross section of this microtubule, you can see this 13 tubulin molecules. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So we have 13 tubulin molecules that are arranged in the cross section. Okay. Now, the next thing that we'll study is where these microtubules are present. So these microtubules are present in cytoplasmic matrix. So microtubules are present in cytoplasmic matrix. Apart from cytoplasmic matrix, these are also present in surface structures such as cilia and flagella. So these are the surface structures in which the microtubules are present. Thirdly, they are present in basal body and centrioles. Basal body and centrioles. And then they are also present in spindle fibers. They are also present in spindle fibers. Now, since all these contain microtubules, then what's the difference between all these? So the difference is the arrangement of these microtubules. Okay, so these all components or these all things have different arrangement of the microtubules that we will see in the later slides. Now let's talk about the function of the microtubules. So what are the functions of microtubules? So the first function is spindle fiber formation. Spindle fiber formation. Now what is spindle fiber? Spindle fiber is a specific type of structure that is used or that has role in cell division that you will see later. But for now, you just remember that the microtubules are involved in spindle fiber formation, and this spindle fiber is involved in cell division. Okay. The second thing is it also involves in anaphasic movement of chromosome. Anaphasic movement of chromosomes. Now, what is anaphasic movement? So when the cell divide, so a cell has two phases. One is the interphase, and the second one is M phase. Okay. And in this M phase, the chromosome division takes place. Okay. Also, this M phase has different types of phases. So 4 phases, that is prophase, metaphase, anaphase, and telophase. So the M phase also has 4 phases: prophase, meta, ana, and telophase. During this anaphase, the chromosomes separate. Okay. So during this, the chromosomes separate, and this separation is known as anaphasic movement of the chromosome. Okay. And this anaphasic movement is also done with the help of microtubules. Now the third is the microtubules also help in providing support to the cells, and fourth, they help in motility. They help in motility because they are also present in the cilia and flagella. Okay, and this flagella helps in the motility, right? So this microtubule help in the motility. Now the next thing that we'll study is about cilia. So we have completed cytoskeleton and the 3 types of cytoskeleton. Next thing that we'll study is cilia. So what is cilia? We have also came across this term when we were studying about the prokaryotic cells. So cilia these are the surface structures. Cilia are the surface structures, right? Now, the important thing is these cilia are only present in eukaryotes. Only present in eukaryotes. So cilia are not present in prokaryotes. These are only present in eukaryotes. In eukaryotes also, eukaryote can be either single cellular organism or multicellular organism, right? So if you see the diagram, so here the first example is of a unicellular organism, and this is paramecium. This is paramecium. So this is a unicellular organism, and we can see the structure here, this surface structure. This is cilia. Okay. So if we talk about a unicellular organism, so the unicellular example is paramecium. Paramecium. Similarly, if we talk about the multicellular organism, so the second figure here is representing the columnar epithelium cell. So the columnar epithelial cells are present in our lungs, and these have this surface structure, surface extension that is called cilia. So we can write about, or we can also say it as ciliated columnar epithelium. Ciliated columnar epithelium. Why? Because it is of columnar shape and it has cilia. So this is known as ciliated columnar epithelium. So in multicellular organisms, the example is ciliated columnar epithelium, and unicellular, it is present in paramecium. Now, what is the role of the cilia? What is the role of cilia? If we talk about the role of the cilia, it act like ore. Now, what does this term ore means? It means it helps in the movement of medium. So a cell has some medium or some surrounding in which something is present, right? And that thing is known as medium. So it helps in the movement of medium and cell. So it either helps to movement of the cell, or it also helps in the movement of the medium. Okay. So if we see the structure of the cilia, so if we draw this epithelial cell, so the structure will be something like this. Okay, so it will be something like this, right? And this is what? This is plasma membrane. The outer structure is plasma membrane. And this is also the extension of plasma membrane, right? It is also extension of plasma membrane. But if we see that this structure or zoom it in the microscope, what we'll see? We'll see a certain structure present something like this in this cilia. Okay. And this special structure is known as axoneme. Okay. So this structure is known as axoneme, and this structure is made up of microtubule. It is made up of microtubule. Okay. So the cilia has this structure that is called as axoneme. Now, next, if we talk about another surface structure, that is flagella. So the flagella also we have studied when we were studying about prokaryotic cells, that it is also a surface structure, right? It is also a surface structure, but it is present in both prokaryotes and eukaryotes. So it is present in both prokaryotes. It is also present in prokaryotes and eukaryotes. Okay. But there are certain differences between the flagella that is present in prokaryote and that is present in eukaryote. So if you remember, in prokaryotes, what we saw, in prokaryotes, flagella has three structures or three parts. One was the basal body. The second was called hook, and the third one was called as filament. Right. So this was the parts of the flagella in the prokaryotes. But if we talk about eukaryotes, so in eukaryotes, it has only two parts. Okay. In eukaryotes, it has only two parts. Flagella has two parts. So suppose this is a cell. So it has only basal body and long filament. Okay. So it has just basal body and filament. So the flagella has two parts in the case of eukaryotic cells, and in this flagella also, in the filament section, they have this structure. This is again known as axoneme. This structure is called as axoneme. Okay. And this is again made up of microtubules. Well, it is made up of microtubules. So what we saw that in eukaryotic flagella, hook is not present, right? Hook is absent. It has only two parts, basal body and filament. Now, if you cut this flagella like this, and visualize it from the top, or if you cut this cilia like this and visualize it from the top, what kind of structure you will see? you will see a structure something like this. Okay, so you will see a structure something like this, and this structure is known as axoneme. So what do we have in this structure? Let's see. So what we can see is the outer membrane here. The outer membrane here is plasma membrane. Okay, the outer membrane is plasma membrane because the axoneme is surrounded by the plasma membrane, right? That we saw here. So this outer layer is plasma membrane. This outer layer is plasma membrane, and the axoneme is present inside. So the outer layer will be the plasma membrane. Now, what other things we are seeing? So the first thing that we can see here is this structure, this circular structure. This is microtubule. This structure is microtubule. And since two microtubules are attached to each other, this is called as doublet. Okay, so it comprises of, axoneme comprises of doublets. That's right. It comprises of doublets of microtubules. And these doublets are present at the periphery, right? They are present at the periphery because this is your cell, this is your cell, and these are present at the periphery. So it is called as peripheral doublets. Okay. So because they are present at the periphery, it is called as peripheral doublets. Now, how many doublets are present here? So we can see the 1, 2, 3, 4, 5, 6, 7, 8, 9. So the axoneme have 9 doublets. Okay. Now, the second thing that we are seeing in the structure is two microtubules are located at the center. These two microtubules are located at the center, and these microtubules are, since they are single, so they are called as singlet. So two singlet are present at center. So two singlets are present at center. Because of this reason, this arrangement is called as 9 plus 2 arrangement of microtubule. Okay. This arrangement is called a 9 plus 2 arrangement of microtubules. So if someone asks, what is the arrangement of microtubules in axoneme? So it's 9 plus 2 arrangement. Okay. Now, apart from this, we can also see this red color structure, right? This red color structure is known as sheath or central sheath. So the red color structure is known as central sheath. And what is the role of this central sheath? So this central sheath basically connects the singlet with the doublet, right? And how it is connecting it? It is connecting it with the help of this radial spoke. So we can see this radial spoke is helping it to connect, right? So we can write that central sheath is connected to doublet by radial spoke. Okay, by radial spoke. And how many radial spokes are there total? So there are 9 radial spokes. Okay, so there are 9 radial spokes in axoneme. Okay, so this is all about the structure of axoneme. Apart from this, one more thing, these doublets are also connected. These doublets we can see are connected by this interdoublet bridge, and then there is a central bridge that is connecting this singlet. So this bridge is connecting the signal. So the doublet are connected by this interdoublet bridge, the singlet are connected by this central bridge. Okay, so this is the overall structure of the axoneme, and this axoneme is present. It is present in both cilia and flagella. In cilia and flagella. And the second important thing is, in this, the microtubule arrangement is 9 plus 2. The next thing that we'll study is about centrosomes. So, what is centrosome? So, if you talk about the structure of a centrosome, the centrosome comprises of two cylindrical structures. Okay. And the centrosome, it is present inside the nucleus, right? So, present inside nucleus. Okay. And it comprises of two cylindrical structures. So, what are these two cylindrical structures, or how do they look? So this is one cylindrical structure, and this is another cylindrical structure, and this is how they are arranged, and they are surrounded by this, something like this. Okay. Now, what these two cylindrical structures are? These two cylindrical structures are called as centrioles. These are called as centrioles. Okay. And they are surrounded by this pericentriolar matrix. Okay. So if you someone asks what is centrosome, so the centrosome, it is a structure that consists of two centrioles, and pericentriolar matrix. And what is the role of centrosome? It helps in cell division. Okay. It helps in cell division. Now, it comprises of two centrioles. So now let's see the structure of centriole. So the structure of centriole looks something like this. Okay. So what we can see here in the structure, first of all, this centriole is also made up of microtubules. So centriole, it is made up of microtubules. Okay. And in this case, what is the arrangement of this microtubule? It is 9+0 arrangement. In the case of cilia and flagella, it was 9+2 arrangement. But in this case, it is 9+0 arrangement. Why? Let's see. So the first thing what we are seeing is, we are seeing these structures, right? These three structures. So these three structures are the three microtubules. Okay. And these microtubules are connected to each other. So this structure is known as triplet. This structure is known as triplet. Okay. So what it consists of? Triplets. And these triplets are present at the periphery or at the side edges, right? So it comprises of peripheral triplets. Okay. Now, this peripheral triplets, how many are there? So if you count 1, 2, 3, 4, 5, 6, 7, 8, 9. So it has 9 peripheral triplets, right? So the centriole has 9 peripheral triplets. The second thing that it has is this central structure, right? This central structure, but it is not a microtubule. Okay. This central structure is called as hub. It is a protein that is called as hub. So the hub is a protein. Okay. So centriole have in the center, in the periphery, it has these triplets. And what are triplets? These are microtubules, right? In the center, what it has a protein called hub. So it has a protein called hub. Now, apart from this, what else it has? We can see that one of the microtubule of one triplet is connected to another microtubule of second triplet by a structure. This structure. And what this structure is called? This structure is called as radial spoke. Okay. So this is called as radial spoke. Okay. Similarly, we can also see that these triplets are also connected to this hub that is present in the center, and this is also called as spoke. This is also called as radial spoke. Okay. So we can write that radial spoke, what it is doing? Radial spoke connects hub to triplet, and it is also connecting triplet to triplet. It connects hub to triplet, and it also connects triplet to other triplet, right? So this is the function of radial spoke. Okay. So in the centriole structure, what we saw that there is 3 microtubules that are connected to each other, and this structure is known as triplet. And there are total of 9 triplets that are present at the periphery of the centriole. And in the center, they don't have any microtubules, but they have another protein that is known as hub. And because of this reason, it is known as 9+0 arrangement. Okay. Now, what this total complete structure is called as, this complete structure, because of the arrangement, the structure is called as cartwheel structure. So it is looking like a cartwheel, right? So because of this, it is called as cartwheel structure. Okay. So the centriole structure is like a cartwheel structure, and it has 9+0 arrangement of microtubules. Next, now we'll study about nucleus. Okay. So what is nucleus? So nucleus is it is a major organelle of a cell, right? If we have a cell, a nucleus, because nucleus consists of a genetic material, right? So nucleus consists of genetic material. And what is the genetic material? It's DNA, right? Now let's talk about the discovery of the nucleus. So the nucleus was discovered by Robert Brown. It is discovered by Robert Brown. Okay. Now, in the year 1831. In what year? In year 1831. Now, how he discovered nucleus? He observed a dense structure in the cell. Okay. He observed dense structure, and that structure was also stained. So dense structure that was stained. Okay. So he observed a dense structure that was stained, and he called that structure as nucleus. Okay. Now, second thing we will see later in detail, but that is chromatin. So the genetic material, which is DNA, in the nucleus, it is not present as a DNA. So the structure that is present in the nucleus, it is called as chromatin. So the genetic material, they fold or they compact in such a form that it is called as chromatin. So what is chromatin? You can write that chromatin is complex of DNA plus protein. Okay. So chromatin is the complex of DNA plus protein, and this complex is very compact. So it is very compact. Now, who discovered chromatin? So chromatin was discovered by Fleming. Chromatin was discovered by Fleming. Okay. And how he discovered it? He discovered it by using a basic dye. Basic means that has base nature. Okay. We have studied about acid and base, right? So he used a basic dye, and with the help of this by using basic dye. Okay. So he used a dye that was basic in nature, and using this dye, he stained the nucleus, and then he observed a structure called chromatin. Okay. Now, if we talk about the position of nucleus in different cells, so according to the cells, the position of nucleus vary. Okay. So if we talk about the plant cells, in case of plant cells, so suppose you have a plant cell. So in that case, the nucleus is present at the periphery. Okay. So it is present somewhere in the side. So this is plant cell. Okay. So in plant cell, nucleus is present at periphery. Nucleus is present at periphery. Okay. But in the case of animal cells, it is present at the center. Animal cells, nucleus is present at the center. Okay. So you have this plasma membrane, and the nucleus is present somewhere here. Okay. Now let's talk about the nuclear structure. But before moving to the nuclear structure, let's see how a cell divides, or what happens during the cell division. Okay. So during the cell division, the first thing that happens is the nuclear division. Okay. So in cell division, the first thing is nuclear division. Okay. So the components of nucleus divide. Now, during this division, the complete cell cycle has two phases. So a cell cycle has two phases. Okay, so suppose this is a complete cell cycle. So it has two phases, a large or time taking interphase and a small M phase, right? So now what happens during the interphase? During the interphase, what happens? In this case, the cell do not divide. In this time, the cell prepare itself, okay, for the division. So it makes the organelle, it doubles the organelle, or it increases cytoplasmic content, but it do not divide, okay. So in this case, cell does not divide, but they prepare for division, okay. They prepare for division. And it is a time where you can see a proper nucleus. So if we want to observe a nucleus in the cell, the best time is to see it during the interphase. Okay. So nucleus is properly visible. Nucleus is properly visible during this interphase. Okay. Now the second phase is M phase. So during the M phase, the actual cell division takes place, right? So cell division takes place during the M phase. Okay. Now in the M phase further, I told in the previous slide also, it has four phases: prophase, metaphase, anaphase, and telophase. So M phase itself has four phases. But in this, when the chromatin or the chromosome is clearly visible, chromatin, just I told that chromatin is a complex of DNA and protein. So the chromatin is visible properly during the metaphase. Okay. So in the metaphase, you can say chromosome. We'll see what is the difference between chromatin and chromosome, but we can write chromosome is clearly visible. So if you want to see a nucleus, then you should see it during the interphase. But if you want to see the structure of a chromosome, it should be seen during the M phase, and in M phase, especially during the metaphase. Okay. Now, let's talk about the structure of the nucleus. So, as we can see, the structure of nucleus looks something like this. So, it has different components. The first of all, because it is a double membrane bound organelle, right? Nucleus is double membrane bound organelle, right? So we can see that it has a nuclear envelope. We can see here it has a nuclear envelope. Now this nuclear envelope, it is also different parts, right? There is this outer nuclear envelope. Nuclear envelope is, it has outer nuclear envelope, outer nuclear membrane or nuclear envelope, you can say. Then it has inner membrane, and also it has perimembrane space, perinuclear space, right? So, or perimembrane space, perimembrane space. So it has this three, nuclear envelope has three components: the outer, inner, and the intermediate, right? Apart from this, what we can see, this is a nuclear envelope. Now, nuclear envelope, there is gap between the nuclear envelopes, right? This gap. And this gap is known as nuclear core. Then, apart from nuclear core, what else we are seeing? We are seeing this condensed structure, right? This condensed structure. This structure is known as chromatin. This structure is known as chromatin. Then we have one central structure, and this structure is known as nucleolus. And then the matrix that is present in the nucleus, it is known as nucleoplasm. So we will see one by one these structures. So the first is membrane. The first structure is membrane. Okay. Now, in the membrane, what we can write, that membrane is made up of lipid. Just like plasma membrane, this membrane is also made up of lipid, and it is bilayer, right? Now, the outer membrane. The membrane has three components. First is outer membrane. So when we are studying the endomembrane system, what we saw that outer membrane is in continuous with one organelle, right? It is in continuous with one organelle, and that organelle is endoplasmic reticulum. So the outer membrane is in continuous with endoplasmic reticulum. And this endoplasmic reticulum, it bears or it has ribosomes, right? It has ribosomes. Okay. Now the second thing is perinuclear space. It has outer membrane. It also has perinuclear space. The membrane has perinuclear space. Now the dimension or the dia of this perinuclear space is it can vary from 10 to 50 nanometer. Okay. It can vary from 10 to 15 nanometer. And it is a space between, it is a space between what? Inner and outer membrane. Inner and outer nuclear membrane. Right. So this is membrane. Now the second thing that we saw was nuclear pore. The second structure we saw was nuclear pore. Now what is nuclear pore? It is the site where the outer and inner membrane fuses, right? So site where outer and inner nuclear membrane fuses. So where the membrane has been fused, that structure is known as nuclear pore. As we can see, right? So here this has fused, right? This has fused, this has fused. Then this structure is known as nuclear pore. Okay. And what is the function of nuclear pore? Through this pore, the RNA that is synthesized in the nucleus and the proteins that are synthesized in the cytoplasm, they move across. Okay. So it is a site or it is a space from where RNA, so from where RNA and protein move. RNA and protein moves. Now the RNA is synthesized in the nucleus. The protein is synthesized outside the nucleus. Okay. So the protein moves in the nucleus and the RNA moves out of the nucleus from this nuclear pore. Right. Now the third structure is nucleoplasm. So what is nucleoplasm? It is matrix of the nucleus. It is matrix of the nucleus. Right. It is the matrix of the nucleus. Now, the fourth structure that we saw was nucleolus. Now, what is nucleolus? It is without membrane organelle. So, it do not have any membrane. Okay. So, it is without membrane organelle. Like what? Like ribosomes. Ribosomes also do not have organelle, right? And what is, if we talk about the number of this nucleolus, it can be 1 to many. So, the nucleolus number varies in a nucleus. Okay. And thirdly, if we talk about the function of nucleolus, it is site for ribosomal RNA synthesis. Ribosomal RNA synthesis. Or you can also write ribosomal RNA as rRNA. So RNA is what? That is produced from DNA by the process of transcription, right? But RNA are of 3 types, among which one type of RNA is ribosomal RNA, that is present on the ribosome and help in the protein synthesis, right? So it help in the protein synthesis. And this ribosomal RNA is formed where? It is formed in the nucleolus. Okay. Now, the last structure is the chromatin structure. So the chromatin, I told that it is a condensed form of the DNA and it also consists of protein. So now let's talk about the structure of the chromatin. So if we see, we said in the initial classes that the genetic material is made up of DNA, right? So DNA, and DNA is what? It looks something like this. It looks something like this. Now, this DNA in the nucleus, it is present in the condensed form. Okay. So it is present in the condensed form. And this condensed form, how it gets condensed? By the help of the proteins. Okay. So it gets condensed with the help of the proteins. So the proteins wrap around it. So there are these proteins, and these proteins are called as histones. So this histones comes and on which the DNA wrap, and they form this structure called as chromatin. Okay. Now, when this chromatin condenses further, so when this become very condensed, something like this, it gives the structure something like this. Okay. So very condensed form of the chromatin, is this structure. But this structure is not always visible. Okay, this structure is visible. It is visible, like I said in the previous slide, it is visible in M phase when they actually cell divides, and in M phase especially it is visible in the metaphase. So this structure is clearly visible, or it is visible in the, this structure is visible in the metaphase of M phase. Okay, so, and what this structure is called? This structure is called as chromosome. This structure is called as chromosome. Now, if you talk about the structure of the chromosome, it has different parts, we can see. So it has two arms. So this is called as, these are two arms. Okay, so this is called as short arm. This is called as short arm. And this is called as, this complete arm is called as long arm. So these are the two arms of the chromosome. Now, these two together are called as sister chromatids. These together are called as sister chromatids. Okay. Apart from this central structure through which these two chromatids are connected to each other, this is called as centromere. This structure is called as centromere. Okay. Apart from this, the edges, this structure, the end of the chromosome, this structure is known as telomere. Okay, this structure is known as telomere. Okay, so this is the structure of the chromosome. Now we'll see what are the functions, or what we can say is what each of these structure means. So the first structure that we see is the sister chromatids, right? So the first structure is sister chromatids. These two sister chromatids. So what we can write about the sister chromatids? So the sister chromatids, they are genetically similar. So what do we mean by genetically similar? Because we know that this is a condensed form, but actually what consists of? It consists of DNA, right? It consists of DNA, right? So genetically similar means that they both have same genetic composition. The DNA comprises of gene, right? So the gene that will be present on this chromatid will be also present on this chromatid at the same location. Okay. And so because of this reason, it is known as genetically similar. Genetically similar. So sister chromatids are genetically similar. And how they are attached? They are attached via centromere. They are attached by this structure that is known as centromere, right? Now, second thing, we have seen the arms, like there are two arms, short arm and long arm. So in one chromosome, so this is one chromosome, this is one chromosome. So in one chromosome, we have two chromatids. This is one chromatid. This one structure is called one chromatid. This is called one chromatid. And similarly, the second structure is called second chromatid. So in one chromosome, we have two chromatids. And these chromatids have two different regions. One is a short region and another is a long region. And these two regions are called as arms, right? Now, the third structure is called what we saw as centromere. What is centromere? Centromere is, it basically connects to two chromatids, right? So it is a structure that connects two chromatids. That connects two chromatids, right? And then telomere. What is telomere? It is end of chromosome. End of chromosome. Okay. So this is all about the structure of the chromosome. Next, we'll see what are the types of chromosome. So types of chromosome is on the basis of, it is on the basis of location of telomere. Sorry, location of centromere. It is on basis of location of centromere. Because we just saw that, suppose this is one, this is two. This is a one chromosome, a chromatid, this is second, and this is centromere. But this centromere can be located at different part or different region of the chromosome. And based on the location of centromere, there are four types of chromosome. The first type of the chromosome is known as metacentric. So what is metacentric? The metacentric, suppose this is your chromosome, this is. So in metacentric, the centromere is located at exactly at the center of the chromosome. Okay. In the metacentric, centromere, the centromere is located at center. Okay. Now the second type of chromosome is submetacentric. Submetacentric. Now in this case, the chromosome is located somewhere away from the center. So it's the center, if you see the center should be this, but it is located away from the center. So we can write away from the center. The centromere is located away from the center. The third type is acrocentric. Acrocentric. So in the case of acrocentric, you can draw a structure. So it is located near the end, close to the end. So this is end. So it is located somewhere close to the end. So centromere is located close to the end. So this is called as acrocentric. And fourth is telocentric. Telo comes from telomere, right? And telomere is the end of the chromosome. So in telocentric, so it will be something like this. Okay. It will be something like this. So here, so this is the centromere. It is located at the end. So at the end of chromosome. Centromere is located at the end of the chromosome, and this is known as telocentric. So based on the location of the centromere, there are 4 types of chromosomes. Okay. So we have studied about the structure of nucleus. We have studied about the structure of chromosome. Now we'll quickly see a very small organelle that is given in the NCERT, and that is microbodies. So microbodies, what are microbodies? They are vacuole-like structure. They are vacuole-like structure. Okay. These are vacuole-like structure, and like most of the eukaryotic organelles, they are also membrane bound. They are membrane bound. So these are vacuole-like structure. But what is special about these vacuoles? That they contain enzymes. They contain enzymes. Okay. So these vacuoles, those that contain enzymes, are known as microbodies. Okay. And these microbodies are present in both. These are present in both plant and animal cells. Plant and animal cells. Okay. They are present in both plant and animal cells. And they contain, so this is a vacuole, and it contains a particular enzyme. And this is why it is known as microbodies. And if you talk about one of the microbodies, the example is peroxisome. So peroxisome is an organelle that looks like a vacuole, and it contains enzyme. And what enzyme it consists of? It consists of catalase and peroxidase. It consists of, these are the enzymes that peroxisome contain. Okay. So this is all about microbodies. And with this, we have completed this chapter, that is cell, the unit of life, in which we have studied about the cell, the prokaryotic cell, and the eukaryotic cells. So that's all. Thank you.